Let's kick off the day with one of our senior researchers, Angela Bownes. Angela is an entomologist specializing in biocontrol of invasive plants. Angela has worked for Manaaki Whenua for almost 4 years now. She moved to New Zealand from South Africa 4 years ago, where she worked mainly on biological control of aquatic weeds. Angela thinks that she might be a moth whisperer, having discovered 2 different species of moth attacking 2 different invasive aquatic weeds in 2 different continents, one in South Africa and another one recently in New Zealand. Angela's talk today provides another view of the current work on weed biocontrol projects funded by the National Biocontrol Collective and MPI Sustainable Food, Fibre, and Future Funds. Angela, if you would like to, please.
Thanks, Arno. Kia ora koutou. Good morning, everyone. So that is a story about the moths. Probably needs a bit more explanation, but that's for another day, or you can chat to me. Oh, sorry. Chat to me during one of the breaks. So as Arno said, I'm going to be giving an overview today of our weed biocontrol projects that we are currently working on for our applied research. That's aims at developing biocontrol agents for release against specific target weeds in New Zealand. As James mentioned, we're also working in the Pacific. Most of that work is funded by MFAT, but today I'm just going to be talking about our New Zealand weeds that we're focusing on. So I apologize in advance. It is going to be a little bit of a whirlwind overview because I've got a lot of ground to cover, which is a good thing because it means we've got a healthy supply of funding for our weed biocontrol projects. But if you've missed anything, would like any further information, please contact me afterwards or chat to me. So just to cover off the funding, so our main funders for our weed biocontrol projects in New Zealand is the National Biocontrol Collective, which is a consortium of regional councils, unitary authorities, and the Department of Conservation. And they're currently funding biocontrol research on 5 environmental weeds. We also have funding from MPI Sustainable Food, Fibre and Futures Fund with co-investment funds from the NBC. So we can't get funding from the SFFF without co-investment funds. And in this case, the NBC is the sole funder of our and that's targeting 6 productive sector weeds. And then more recently, we have funding from 3 individual regional councils that are interested in developing biocontrol agents for weeds that are particular issues in their regions, but are not an issue on a national level. So they're not funded through the National Biocontrol Collective. So kicking off with banana passion fruit. Banana passion fruit are the weeds that we're focusing on in this are the Passiflora species in the Taxonia subgenus. They are large perennial high climb. Sorry, we've got a bit of an interesting situation here. I've got to work with 2 things. I forgot. Hopefully I won't going forward. Okay. So yeah, so I'm sorry. Okay. So banana passion fruit. We are targeting Passiflora species in the Taxonia subgenus. They are large perennial high climbing vines that smother canopy, forming large masses, and they outcompete native plants and prevent regeneration. So for this project, we're focusing on 2 candidate biocontrol agents. We haven't actually imported them into New Zealand yet, unfortunately. We've been battling for many years now to get an export permit from Colombia, which is where these 2 agents are, candidate agents are native. We do have a subcontract agreement with the university in Colombia. We've had a master's student working on this project for the last couple of years, and she's done some preliminary research for us on these candidate agents. Sorry, this is going to take a while to get used to. Please just give a prompt. So on the left, we have a stem-boring moth. As the name implies, it attacks the roots of the stems of banana passion fruit. And then on the right, we have a seed-feeding fly. So the student has done some work on, particularly on the stem-boring moth. One of our native Passiflora species, Passiflora tetundra, has been planted out at a field site in Colombia, and she's been monitoring that for non-targeted for attack on that native plant. So that's given us an idea of host specificity. So over the last few years, there's been absolutely no signs of attack, which is a good thing. So it's looking to be really host specific. You're probably wondering about New Zealand native plants being planted out in the field in Colombia. It's a dioecious plant, so only one sex has been introduced there, and so the plants don't flower. That does also mean we haven't been able to look at the effects of the seed feeding fly on this species, so we don't know much about the host range. But so these are two promising candidates, but we have the export issue from Colombia to resolve first. The student has also developed an artificial diet for the stem-boring moth, which is really good for mass rearing and building up numbers for testing. So then on to Darwin's barberry. Darwin's barberry is an evergreen spiny wooded shrub. It's mainly a problem in Southland, Canterbury and Taranaki and Greater Wellington. It has long-lived, well-dispersed seeds, replaces shrubland and regenerating forest, and is a threat to forestry, pasture, and native ecosystems. So we do have one biocontrol agent that has already been released against Darwin's barberry. That's a seed-feeding weevil. We're seeing good signs of establishment down south, but it hasn't really established well anywhere else in New Zealand. So we're still doing some work on that agent. But the agent that we currently have in containment here in New Zealand that we're hoping to release is a rust that damages the leaves and the fruits of Darwin's barberry. So this project was also has also been delayed. Darwin's barberry is native to Chile. So COVID had quite a dramatic impact on our ability to progress this research for a couple of years. It's a very difficult agent to work with in containment. It's slow to build up spores for testing. So we were regularly collecting spores, fruits, infected fruits and leaves from the native range to do our host range testing. So COVID caused some delays, but our technician on the project, Chantal, was back in Chile in December last year, and she's got enough inoculum to complete that host range testing. It's looking to be really host specific, and we're hoping that we'll be ready to start preparing a release application for this agent next year. Next is Japanese honeysuckle. I'm sure most of you are familiar with Japanese honeysuckle, a widespread, very big problematic weed in New Zealand. It's an evergreen climber. Again, a climbing smothering habit. It smothers plants from ground to medium canopy, forming dense mats and smothering native vegetation. So we do also for this project, for this program, we have one agent that's already been released. Some of you might be familiar with the beautiful butterfly, the Honshu White Admiral. We were mass rearing that until recently. Last year was our last season was our last year for supplying releases to the regional councils. And now we're focusing on this stem boring cerambycid beetle. So this has been approved for release. We've been mass rearing it in the labs to conduct releases mainly in Hawke's Bay. We've also done some releases at our site down in Lincoln. It's been a difficult agent to work with, being a stem borer, and it's got a long life cycle. It's difficult to keep the plants healthy under glasshouse conditions. So our rearing efforts have been a little bit disappointing. We were expecting to get higher numbers out. We do have signs of establishment at our Lincoln site. And if it's got a 2-year life cycle, we're still unsure of whether it's a 2-year or 1-year life cycle. We're expecting to get high numbers out this next season. If we don't, and also our mass rearing cultures at our lab in Tamaki don't produce high numbers, we may consider going back to the native range to recollect the beetles and try and rear them through one generation, get high numbers out into the field. But if we can get it established, it's a really damaging agent. So we have high hopes for this, even though it's been a little bit of a challenge to work with in containment. Lagarosiphon major is a submerged oxygen weed, also distributed throughout New Zealand. It forms dense mats, smothering native aquatic vegetation and reduces the availability of oxygen in the water, which impacts fish and other organisms. And for this weed, we've been working on 2 candidate agents for the last few years. So a lot of the research on these 2 candidates was done in Ireland in the early 2010s. They also have a major issue with Lagarosiphon there. Unfortunately for them, before they completed all their testing on this leaf mining fly and a shoot mining midge, they ran out of funding and they weren't able to get any further financial support to actually go ahead and release them. For us, we were fortunate to be able to piggyback on that research while they had cultures of these 2 insects in their containment facilities. We were able to get them to test some of our native plants on the fly, which is the one on the left. So that work's been completed. It's sufficiently host specific for release in New Zealand. So that's just kind of been on the back burner while we've been focusing on the midge, which is the bottom left there. So that's some for the agents, some preliminary research was done in Ireland, but they didn't complete the testing. So we contracted some collaborators at a university in South Africa to complete the testing of the midge for us because Lagarosiphon's native to South Africa. And unfortunately, COVID, droughts and floods have prevented our collaborators from collecting a field colony of the fly to get a colony established in the lab and to do the testing for us. So we've had severe delays with that, and we do want to apply for permission to release both agents at the same time to EPA. So that this project's unfortunately been stalled for 2 to 3 years now. But they're continuing to try, and unfortunately this midge, it's got a very limited distribution in South Africa. It's only known from a few field sites. So until those field populations recover, we can't progress that project any further. So moth plant, a nasty weed in the Auckland area and Northland, Waikato. It's another climbing vine. You'll see that we're working on a lot of vines and climbers, and the reason for that is because they're very difficult to control with other conventional methods. With chemicals, obviously, they target the underlying vegetation also gets damaged. So biocontrol is really the best option for managing vines. So moth plant produces a milky latex in the stems, leaves, and roots, and it's again smothers native vegetation, preventing regeneration and recruitment, and it's got the potential to impact forest structure. So for moth plant, we've been working on 3 biological control agents or candidate biological control agents. The one on the left is a root feeding beetle. The adults feed on the leaves, but the larvae attack the root system. They're the damaging life stage. That has been approved for release. It was released in 2018. We're seeing really, really good early signs of establishment and damage in the field. And we are still continuing to mass rear this agent at our lab in Tamaki to provide to the regional councils. The one in the middle is a fruit fly, a wasp-looking fruit fly. But you can see in that picture there, the larvae feed inside the pods and they damage the seeds and completely turning them to mush. So we have really high hopes for this agent. But the EPA application process has recently been stalled. We submitted an application. It went out for public consultation, and unfortunately during that process, MPI picked up that we have trading agreements with key trading partners that have a blanket ban on Anastropha species. So this fly is Anastropha australis because there are 9 species within that genus that are of economic importance, and particularly one in the Curvicorda species group is a pest of fruit papaya. So we've requested a time waiver from the EPA to put a pause on that application process while we negotiate with MPI to resolve those barriers. We had our first meeting last Friday. We're very positive. MPI is very supportive of assisting. They've got to, I guess, make the changes to these trading agreements. They're very supportive of that and going to work with us. So we're confident that it's going to be resolved. We just don't know how long it's going to take. And then for the last agent is a rust fungus. We actually have approval to release this rust fungus that was granted by the EPA in 2015, but we haven't been able to get an export permit from Argentina for that agent yet. Also, again, a work in progress And we've got collaborators in Argentina that are doing some other sort of work on this agent on the side. So it's in the pipeline, but until we get that export permit, we can't progress that. Challenging work it is, but we do have some successes, many successes. So next up is wild ginger. So this project is also kind of on the back burner. There's not much work currently going on. We've been working with a collaborator in the UK who's done some testing for us. They've managed to do testing on 2 agents, quite extensive testing, but they lost the cultures and they haven't been able to get renewed permits to collect the agents out of India. So that program is also on a little bit of a stall, but just to cover wild ginger, it's a non-woody perennial herb. Forms dense stands through the forest understory and on the margins of waterways and roadsides, smothers and replaces all understory species and seedlings. So the 2 agents that our collaborators at CABI have been working on is a little fruit fly, which is the one on the left, that's a stem miner, and the one on the right is a rhizome feeding weevil. So they did quite extensive testing with the fruit fly, F-R-I-T, it's not a fruit fly. And unfortunately, after doing extensive testing, they found out that they had 2 species of fly, cryptic species of fly in containment rather than 1. And then there was no way to separate which species they'd done the testing on which. So that was, an unfortunate delay. And they haven't been able to import that agent, get that agent out of India again. And the same with the rhizome feeding weevil. They've done some preliminary research, but lost the cultures in containment, a really difficult insect to rear. And they've been having issues with the Indian authorities to get approvals to get the insects out of India again. But we have been talking to collaborators. We just come back from an international conference. We've been talking to collaborators that maybe are able to assist with that process, or there's potential to look for these agents elsewhere. So that program's kind of on the back burner, but such a high priority weed and we've spent a lot of money already, or the NBC is and SWF invested a lot of money in this project, so it's still worth pursuing. Then Chilean flame creeper, another climbing perennial vine. It invades forest margins, disturbs sites. It's a particular problem in Southland and becoming more of a problem in Canterbury and up north. Climbs into tree canopies, suppressing growth, replacing native species, and through smothering and shading. So the agent that we're working on for Chilean flame creeper is this leaf-feeding beetle. So this was a serendipitous discovery, an opportunistic discovery when Chantal, our technician that's working on Darwin's Barberry, had gone over to Chile to go and collect more of the inoculum for the rust. And they found Chilean flame creeper plants and did a little survey, and they picked up this chrysomelid beetle, which make really good biocontrol agents. So with that finding, it was kind of bumped up the list as a good priority, a good target, as an easy agent that we could work with. So we imported this agent in November last year. My colleague Ronnie Gruntman, who's leading this project, was very organized, got all the test plans lined up, and managed to complete the host range testing for this beetle within a few months. So this is one of the projects that we've had really good success in a short space of time. Things have moved much faster than we would normally expect. So really exciting for that. And Ronnie's analyzing the data, and we're hoping to submit an application to the EPA. It's looking really host specific. We might do some more detailed testing, but it's looking really good. And we're hoping to submit a release application to the EPA next year as well. Chilean needlegrass, I'm not going to spend too much time talking about Chilean needlegrass because, this is Chilean flame creeper leaf beetle. Chilean needlegrass, I'm not going to talk too much about it because my colleague Alana Dembrian is talking about Chilean needlegrass, but it's a major pasture weed. It's got these horrible seeds that have a needle-like tip that are harmful to livestock, and it downgrades the hides in meat and wool. I borrowed this map from Hawke's Bay's website because the iNaturalist, oh my goodness, 2 minutes. The iNaturalist the map was not accurate at all, I think, because grasses are so difficult to identify, so people aren't putting locality data up there. But we've got a rust fungus that's been 10 years in the making, so Alana will talk about that. I won't talk much about that. So Old Man's Beard, I'm sure you all know about Old Man's Beard. It's a massive weed throughout the country, another climbing layering vine that smothers and kills native plants and preventing regeneration. So we're so we're working on 3 agents for Old Man's Beard. On the left is a sawfly that Arno is working on. This agent was released quite a few years ago, 20 or 30 years ago, and we have revisited it because we had really limited establishment in the field. So this time we managed to mass rear thousands and thousands of larvae, and we decided to put all eggs in one basket, and they were only released at a site in Amberley. Arno's been monitoring that very closely for the last few years. And we're starting to see they've definitely established, starting to see really good signs of damage. So now we're exploring ways to redistribute this agent and to mass rear it in the lab to supply to regional councils so we can get it spread around the country. The Old Man's Beard mite is one in the middle. We are currently mass rearing that agent for releases, getting it around the country. We have high hopes. It's established really quickly and it's really good at dispersing itself. But no, it's still too early for impact assessments or impact to be visible. And then we're also working, we've got collaborators in the UK that are working on another pathogen for us. We did release a pathogen also decades ago when Old Man's Beard was first worked on. But the pathogen died out, so we're now revisiting other options, and some promising candidates have been identified, and our collaborators are doing pathogenicity studies to see if they're infectious against our biotypes of Old Man's Beard. Sydney golden wattle is a woody shrub, small tree. It's particularly a problem in the dunes of Manaaki Whenua and up in the Kamama swamps. And this is our latest edition. Our latest edition is this little flowerbud galling wasp. So it's a herbivorous stingless wasp. So we got approval to release this in November last year. We did the first releases. It was already in the country while we were waiting EPA approval in containment and MPI approval. And we have high hopes for this agent. It's been a really successful agent, a biocontrol agent in South Africa, and more recently it was released in Portugal, and they're seeing very, very good signs of damage. So yeah, this was a welcome addition to our biocontrol agent group, and yeah, it forms these instead of the flower. So it lays eggs into the primordial buds of next that will turn into flowers or vegetative buds or growth following season. And so instead of producing flowers, it produces these galls, which is pretty cool. Woolly nightshade is a shrub or small tree. It's mainly a problem in the North Island. It crowds and shades out native plants and inhibits native forest regeneration. We are currently working on a flower bud weevil in collaboration with Wits University in South Africa. It's a high priority weed for the South Africans as well. So it's a little flower bud feeding weevil. They've done extensive research on several native solanum species and economically important solanum species. For us, all that we need to test in conjunction with what they're testing are our porta species, Solanum viculare and Solanum laciniatum. So we've made several attempts to try and get seed over to South Africa so that they can grow the plants and test for us. And unfortunately, they haven't had any luck germinating those seeds in the lab. And we've also had difficulty locating field populations of Solanum viculare to collect seed. So this program has also had its challenges, but we've managed to locate a few bushes on Banks Peninsula this last season, and the fruits are currently maturing, and we're hoping that we'll get viable seed out of that we'll be able to send over for testing. Then also on the right, we've got a few other agents in the pipeline that we are interested in potentially importing and studying, but we need to get to South America and get permits to survey to look for. Those are stem gallers and stem boring weevils that would be really damaging. The yellow flag iris, it's an aquatic perennial iris. You can see what kind of impact it has on waterways, and also the seeds are poisonous and may have an impact on wildlife. Again, for this project, we're working with collaborators in South Africa. Yellow flag iris is also a major weed there. They started up a program a number of years ago, and we are piggybacking on them. So they've done extensive testing with this flea beetle on the left that damages the leaves, and the larvae mine the leaves of the plant, and the adults strip the epidermis of the leaves off, like you can see in that picture. The important species for us to test are native Lagarosiph species. They have also had trouble propagating these species under containment conditions where the flea beetle is. So we've tried various things. We're sending seed again very soon as the last attempt to see if they can propagate those species for us and do some testing. And if that fails again, then we will look at importing the flea beetle and doing the testing here so we can get the Lagarosiph species tested. Looking like it's an iris specialist. So we think it will be host specific enough. We don't have any native iris species. It's really just the Lagarosiph that we're worried about. Well, we'll need to consult with the Iris Growers Association, of course, in that process, but and then on the right, we had high hopes for this little weevil. It's a seed-feeding weevil, but more recently at this international conference, we had a meeting with our collaborators there, and it turns out that some field work they've done, it looks like it has a broader host range than the flea beetle. So, and it also, the adults feed on the flowers. They don't only attack the seeds. Well, the larvae are inside the seeds, but the adults feed on the flowers. So we're probably not going to be able to pursue that one, but the South Africans are still looking at it. So we'll just wait to see what their research shows. But it's probably not going to be a candidate anymore. Then purple loosestrife. So sorry, purple loosestrife. This is a project funded by Horizons Regional Council. It's naturalized in Canterbury, but it's mainly a problem in Manaaki Whenua, and they don't have any tools to manage it. So they're exploring biocontrol. Their 4 agents were released against purple loosestrife over 30 years ago in the USA to leaf-feeding weevils and 2 weevils, leaf-feeding beetles and 2 weevils, a root-feeding weevil and a flower-feeding weevil. And we don't have any native close relatives of purple loosestrife in the family Lythraceae. So we have proceeded with a release application with our testing. We're using the testing that was done in the US and field evidence of their host range, these agents' host range, and we are hoping to submit that release application within the next couple of months to the EPA. Then I'll just run through these. So evergreen blackthorn, this is a major priority weed for Auckland Council. We've completed a feasibility study on this weed, and the next step, we have a number of steps in our weed biocontrol projects. First is always a feasibility study, and then we do native range, New Zealand surveys, just to be sure that we don't have any of the agents that we're interested already here. There's not something already present on the plant that could contribute to managing it. So we are currently undertaking those surveys now. We've just done a batch of autumn surveys, and we'll be doing some summer surveys next season, and we'll see where Auckland Council wants to take, if they want to take that any further. And then lastly, this is a brand new potential project, Madagascar ragwort. It's a big problem in Northland, mainly just for Northland, but it's a scary weed because it's a productive sector weed, and it's also poisonous to horses and livestock. So there's a program going on in Australia for this weed, and Northland Regional Council is interested in piggybacking on that. So we've got funding now to do a feasibility study through an EnviroLink grant, and that will be the first step in this program to see whether biocontrol is a feasible option, and while we watch what's happening with the program in Australia. And that's my whirlwind overview of our weed biocontrol projects in New Zealand, and just to acknowledge all the funders and collaborators.